Hi everyone, in this video we are going to be talking about diffusion and osmosis. And we'll get to the technical definition of these words in a few slides, but essentially what we're going to be covering in this video is water balance and regulating water balance. And so when we're thinking about why water balance matters, one reason is that water is going to make up the majority of many organisms' body weights. So for humans, I think we're about 60% water, and our blood is even more, like 90% water. So if you think about all of the watery cytoplasm that is in the inside of our cells, that's going to be a big component to our bodies in general. Additionally, oceans cover about 70% of the Earth's surface, so there are a ton of organisms that live in the water and that have to be able to regulate how much water is on the inside of their cell membranes versus the outside. Finally, water is involved with a ton of functions. So there's a list of them here: regulating body temperature, transporting nutrients, cushioning organs, flushing out toxins, providing support in plants. We need water for metabolic reactions, so photosynthesis, metabolism of fats, and it's also involved in digestion. This is not even a comprehensive list, but basically, in order to do any of these and other functions, we need water. So before we get into diffusion and osmosis, I just want to cover a couple of vocabulary words that we're going to be using. One is the word solute. And solute is going to be a substance that's dissolved in some kind of liquid. And we call the liquid the solvent. And when you mix a solute and a solvent, you get a solution. So solute is the thing being dissolved. Solvent is the substance in which the solute is dissolved. So an example would be salt could be the solute, water could be the solvent, and then we get a solution of salt water. In biology, most often the solvent is going to be water. So we'll have all kinds of different molecules dissolved in water. Okay, so let's first think about which direction solutes will move in if they're free to do so. In general, particles are going to move from where there's more of them or where it's more crowded for the solute to where there's less of them or where it's less crowded for the solute. And this idea is called diffusion. So, diffusion is the passive movement of particles from regions where they're highly concentrated to regions where they're in lower concentrations. And you can see this visually here, where if you dropped food coloring into a beaker of water, the food coloring will spread out from where it's more concentrated in this drop here, and then it'll keep spreading to where it's less concentrated until there's sort of a uniform distribution of the dye molecules. And you can see it on a little bit more of a molecular level here. So here we're using dye molecules as the solute, they're the things being dissolved, and the solvent is water. And so when we're thinking about where these solute molecules or dye molecules will move, they're highly concentrated down here, and so they're going to move out to where they're less concentrated, so up towards the top of the beaker. So the dye molecules or the solute will move up and out, and the water molecules are also going to move into the bottom of the beaker where they're less concentrated until we get an equal distribution of molecules. So again, diffusion is just molecules moving from where there's more of them to where there's less of them. So you'll notice in this diagram down at the bottom that we showed the solute molecules moving, the dye molecules moving, but we also showed the water molecules moving. So these blue arrows here are showing water molecules moving from regions of high water concentration to regions of lower water concentration. So we can think of the movement of water molecules as being sort of a special case, and it happens so often in biology that it actually gets its own special word. So, osmosis is the diffusion of water across a semi permeable membrane. And water is going to move in the same way as any other particle. So, if you look at this top diagram up here, this dashed line is supposed to represent a membrane, for example, like our cell membrane. And all these little blue circles here are water molecules. And they're going to move from the left side to the right side. And that's because on the left, there's a higher concentration of water molecules. They're more crowded, there's more water over here on the left, and there's less water on the right. So water is going to move from a high concentration of water to a lower concentration of water. So it's basically just a special case of diffusion, like the exact same idea that we talked about on the previous slide. The one thing that can get a little confusing is that we can also talk about water moving from a lower solute concentration toward a higher solute concentration. So, what we're thinking about in terms of when we're thinking about water moving in relationship to how many solutes there are, if you look at this top photo or top diagram, there are fewer solutes. So, that would be these red molecules that are dissolved in the water, fewer solutes on the left side, and a greater number of solutes on the right side. So, as water is moving from the left to the right, it's moving from where there's more water to where there's less water, but it's also moving from where there are fewer solutes to where there are more solutes. So, water moves from high water concentration to low water concentration, but it also is going to be moving from where there are fewer solutes to where there are more solutes. So this is shown over here on this bottom diagram as well. This orange layer is a membrane, and on the right-hand side we have pure water, and on the left-hand side we have a solution. And so the water concentration is going to be higher on the right and lower on the left. So water is going to move from right to left, but we could also think about it in terms of solute concentration. And the solute concentration is higher on the left, and water is going to move in that direction towards the higher solute concentration. Okay, so when we're thinking about the movement of water, a related concept is this idea of tonicity. And this is going to refer to the relative concentration of solutes on the inside versus the outside of a cell. And we have three kind of conditions that we can consider. So, one condition is when there is a hypertonic solution. And this is when there's going to be a higher solute concentration on the outside of the cell, and water will follow that higher solute concentration out, so water will leave the cell. In the middle, we have the idea of an isotonic solution. So this is when we have the same solute concentration on the inside and outside of a cell. So this is going to give us an equilibrium where water is entering and exiting, but the net movement of water is going to stay the same. And then finally, we have a hypotonic solution. And this is when there's a lower solute concentration on the outside of the cell. And so water is going to come into the cell towards the higher solute concentration on the inside. And you can see these three conditions represented in diagram form here. So the first one, hypertonic, remember that's when we have higher solute concentration on the outside of a cell and lower solute concentration inside. So water is going to follow that high solute concentration and it'll leave the cell. Isotonic is when we have equal solute concentrations inside and outside. So water will be both exiting and entering, but it'll be doing so in equal amounts. So there's no net movement of water. And then finally, a hypotonic environment is where there's less solute outside the cell and a greater concentration of solutes inside the cell. And again, water will follow from where there's less solute concentration to where there's a greater solute concentration, and it'll enter the cell. So, this next slide, we're going to be thinking about how these different conditions, hypertonic, isotonic, and hypotonic, affect both plant and animals. So, again, here we're going to start with hypertonic, and that's when there's a higher solute concentration outside of the cell, so water will leave. And in an animal cell, we're showing as our example here a red blood cell. Water will leave, and we're just going to lose a bunch of water on the inside of the cell, so the cell's going to shrivel up. On a plant cell, it's going to have that rigid cell wall on the outside. So water will leave, and the plasma membrane or the cell membrane will sort of shrivel away from that cell wall, but it won't completely shrivel up like an animal cell would. So the plant's still going to be very unhappy. It's going to be very wilted and sad looking, but it's not going to shrivel in the same way that an animal cell would. Isotonic is when there's no net movement of water. Water is going to be both entering and leaving because the solute concentrations are the same on the inside and outside. So animal cells are going to be happiest in this condition where there's no net movement of water. Plant cells are going to be okay. They're going to be a little wilted, but not nearly as bad as this hypertonic environment. And then finally, hypotonic is when there's a higher solute concentration on the inside of the cell. So maybe there's like pure water on the outside. Then water would come into the cell. Animal cells, since they don't have a cell wall, there's just a cell membrane, they'll get really swollen and eventually they can actually burst open, and that's called lysing. So animal cells are not happy in hypotonic environments. But plant cells, because again they have this cell wall, water can come in and it'll be really like full, make the central vacuole really full, sort of make the cytoplasm extra plump, and the interior contents of the cell are going to just push against the cell wall, and it'll actually just make the plant very um, upright and sort of perky. So a well-watered plant is going to have these turgid cells. So plant cells are happiest in hypotonic environments. This last slide is getting at this idea of osmoregulation. And this is the idea that um, many organisms, all organisms basically, are going to have to control how much water and dissolved solutes they're gaining or losing depending on their environments. So there are two examples shown here. On the left, we're showing a paramecium, and these are single-celled organisms that live in watery, freshwater environments. So since it's fresh water, there's going to be more solutes inside the paramecium than on the outside. So water is going to want to come into the cell. And we don't want this organism to end up like our red blood cells, right? We don't want them to burst open. So they have what are called contractile vacuoles. And essentially, the water is going to come in and it's going to enter these vacuoles, and they are going to swell up and fill up with water. And then the contractile vacuole moves to the edge of the cell and sort of just dumps the water out. And then we just keep doing this to regulate the amount of water inside of this paramecium. And then on the right-hand side here, we have fish, and fish can live in both freshwater environments like streams or rivers, and also in the ocean where it's really salty. So the strategies that these fish will need to use in order to maintain the right levels of water and solutes inside of their bodies is going to depend really strongly on which environment they're in. And this picture on the top is showing a fish in fresh water. And so fresh water is basically pure water. And so the fish is in a hypotonic environment. And so water is going to be trying to come into this fish through the skin, the gills, through the mouth. Water is trying to pass any membrane that it can into the fish. And so in order to combat this, the fish is not going to be drinking very much water because it's already got so much coming into it. And then it's basically going to pee a lot. So it's got really dilute urine that's basically all water. And so it's just trying to excrete this very, very dilute urine to get rid of that extra water. And then on the bottom, we have a fish in a saltwater environment. So this is going to have completely the opposite strategies. So it's going to drink a lot of water because water is going to be trying to leave towards that higher solute concentration in the ocean. And then instead of excreting dilute urine, it's going to have really, really concentrated urine because it can't afford to lose any excess water. So, osmoregulation is going to be an important process for pretty much every living organism. And this concludes our lecture on diffusion and osmosis. So, I'll see you guys soon.